Hello, everyone. Welcome to the H3A BioNet webinar for the month of October. My name is Chinakaho Malesa, and I'm a training coordinator with H3A BioNet. Today, we have with us Dr. Alfred AmambuaNgwa. I hope I said that right.
Oh, that sounds great.
Thank you. He will be talking to us about, he will be giving a presentation titled From the Continent to Households, Malaria Parasite Lands, Interconnectivity and Survival Strategies. I'm just going to give a 2 minutes to allow more people to join. And while I do that, I will go ahead and read his bio before Dr. Alfred gets started. So Dr. Alfred is an associate professor from the Gambia. He is with the Disease Control and Elimination theme at the Medical Research Council Unit, the Gambia at London School of Hygiene and Tropical Medicine. He's also an international fellow at the Wellcome Sanger Institute. Dr. Alfred has obtained his BSc up until his PhD in biochemistry at the University of Buea in Cameroon. He joined the MRC in 2006 and has been engaged in cell biology, microarray, transcript analysis, and genome sequencing to determine the diversity in erythrocyte invasion patterns and genetic signatures of selection of the deadliest malaria parasite. And that is what his talk will be focusing on today, and I'm sure you'll get to hear more about it as he speaks on this. Dr. Alfred, when you're ready, you can get started. Thank you.
Yeah, thank you very much, Tina, for inviting me to do this webinar. I can see that I already have a lot of converted disciples that are listening to this for the nth time. But permit me start by going soft on the presentation and also presenting some of the things about malaria that might be important to set the stage to understand what we're talking about here. So, since 2008, part of the work that we've been doing in the Gambia is to try to understand the genetic diversity of the parasite, not only in the Gambia, but also to work with colleagues across the continent together as a team to further understand the parasite and contribute knowledge that might help in refining some of the elimination strategies against malaria. So, just to give an introduction, malaria remains a significant problem not only in Africa, but mostly in Africa. It is caused by 5 species of parasites. One of them is a zoonotic parasite that is mostly common in Southeast Asia, but the other 4 species can be found in Africa. And this includes falciparum, vivax, ovale, and malaria. The deadliest of all of these is the Plasmodium falciparum, and it's responsible for most of the infections and deaths that are recorded annually from malaria in Africa. The parasites, as they're different, they also explore something unique, which is something that is explored by some protozoa. They use a vector to jump from one individual to the other. And that makes it quite complicated because there are 2 hosts. There's a human host and a vector host. Now, just like with humans, though we are not diverse by species, we're diverse genetically. But for the vector, there are several species of the vector that exist across the continents, across the world, and the efficiencies in transmitting the disease do differ. And so to tackle the disease, you not only have to tackle the parasites in the human host, but also have to tackle the jumping of the parasite from humans to the vector, and have to tackle several different types of vectors across different places where the disease is endemic. So at the moment, malaria control strategies and elimination strategies, they include several different tools that can be categorized into several different approaches. This includes diagnosis, for example. I think it's quite important that most of the infections are detected early enough and such that treatment can follow. And luckily enough, there are rapid diagnostic tests that are available to allow for rapid detections of infections using RDT. but these also suffer from problems. They suffer from problems of sensitivity that might be caused by genetic factors in the parasite, such as deletions of the genes that are important for the detection of the parasite when they infect humans. The second strategy, obviously, is treatment. In treatment here, we're talking about use of drugs. And there's been several drugs that have been developed against malaria, and these have been used over the centuries. Again, these drugs suffer from one common problem. The parasite is good at adapting to the drugs, and they develop resistance to drugs quite quickly. And so there have been changes of drugs over time, drug policy over time, and currently it's combinations of drugs that are being used based on the adaptation needs. And these combinations of drugs are quite effective. But resistance as well is also developed to the drugs. It's developing to these new combinations of drugs, as has been the case for the old ones. So there is need for more drugs to be found and to understand how the parasite responds to these drugs. Then, of course, there are other preventative measures that can be taken. And most of these can be directed at the vector, to stop the vector from biting humans. So essentially having a barrier between vectors and humans using mosquito bed nets, for example, or using repellent that would stop the mosquitoes from feeding on humans. There is also mass insecticidal treatment, such as the spraying of houses or larviciding of breeding sites. Now, these methods are not cheap, and so they suffer from issues of cost, and the mosquitoes as well have also developed resistance to the insecticides that are being used. So besides the problem of drug resistance, there is also insecticide resistance that has to be handled. And this is also genetically linked. So there are markers, mutations that have occurred in the mosquito population that have allowed them to survive against the various insecticides that have been developed, and even the new ones that are coming up recently might also face the same problem. The last tool that might be employed for control of the disease could be a vaccine. There is no real wide-scale vaccine available for malaria at the moment. There is the RTSS that has been developed, tested, and proven to be efficacious, and that could be used in some countries. And actually, the WHO has given the approval for it to be used in some countries. There is a new version of the vaccine coming up called the R21 that could also be widely used and has been shown to be highly efficacious. But at the moment, there is no vaccine that is cheap and available for use across the entire world. Despite what I've just said, the combination of all these tools over the last several decades have actually resulted in a significant decline in the malaria incidence and deaths across the world, and including Africa as well, where most of the cases of malaria, especially that from P. falciparum, are found. So there has been a decline, for example, in our country, the Gambia, of over 50% of incidence in the last 20 years. But in the last 5, 6, 7 years, there has been a plateau in that decline in the incidence. And even now, there's an indication that there's an incline, which is an increase in the incidence of malaria in some of the countries where we had previously thought that malaria has declined significantly. And last year alone, there was over 600,000 deaths from malaria in 2021. Now, part of this could be due to COVID, but it could also be part of the process of the parasite, as well as the ecology, giving the parasite the opportunity to survive and continue to transmit in populations. Unfortunately, as it is called the disease of poverty, 95% of the incidents and cases and deaths are found in Africa. And this is a significant problem because the African countries don't seem to have enough resources to combat the disease. Now, there are some problems beyond the strategies that are being used, beyond the insecticide resistance. There are some other factors that might be contributing to the persistence of malaria in our populations, including Africa. Africa is a developing continent, so you have new metropolises emerging, and that means new dense populated areas in cities with probably poor drains that may allow for breeding of the parasite vectors. There are new farmlands and marshlands that are flooded, and there's climate change with new floods all over the place, and also poor housing, which is a problem with development. So all of these are also contributing to the persistence of malaria in our population. An example which is actually intriguing and important is the identification of Anopheles differensii in the Horn of Africa. And recently, there are indications that in some parts of West Africa, Anopheles differensii might also be present there. And this is very efficient in transmitting urban malaria. And if that be the case, it would become more difficult to actually eliminate the parasites from our population as is planned. Now, the WHO has actually developed a strategic framework for elimination of malaria from the populations. This was adopted in 2015 and 16 by the various core groups, and there are three pillars in this technical strategy for malaria elimination, with the hope that by 2030 malaria will be completely eliminated, hopefully, from the rest of the entire world. We are about 7 years or so from there. And I'm looking at that and I'm wondering whether we need to go back and do a new strategy. There are two things that I pick up in that strategy and that we as a group, we thought that we can contribute to. It is to the third pillar, which is to transform malaria surveillance into a core intervention. And we can combine this with one of the supporting elements, which is harnessing innovation and expanding research to be able to provide information that will help the NNCPs to better control for malaria. What we have focused on has been the genomic surveillance. And we'll focus on genomic surveillance because we've had the luck as a consortium of scientists across Africa to have very strong interaction with the malaria gene at the Wellcome-Sanger Institute, giving us the facilities to be able to interrogate the genetic diversity of parasites collected from clinical and community cases. And this has been made really easy by the fact that we can now sequence parasites from filter papers and collecting samples and specimens from infected individuals using FTA cards or using Wadman filter papers is pretty much easy. And that has facilitated the generation of tons of data from the genome across different countries under different conditions that we are currently exploring. So the rest of my talk is going to be about the work that we're doing, looking at the genetic diversity of the parasites from the genomes that we have actually collected. Now, this work is done by a consortium of individuals that belong principally to the Plasmodium Diversity Network in Africa. That's how it was called, the PDNA, but it has evolved now to become the Pathogen Genomic Diversity Network Africa, so that we are able to explore our skills beyond the malaria parasites and look at other pathogens using similar genomic approaches. So the PDNA has been very instrumental in studying the parasite across Africa by collecting parasites from different countries. I think as of the last count, there were 17 countries, but this is growing to include many more malaria endemic countries across the continent. So one of the first questions that we wanted to ask of the PDNA is about the complexity of infection across different countries. And we know quite well that the vectors differ, the human populations differ. The ecology of the different settings where malaria is endemic also differ. And in some places, malaria is perennial, while in other places, there are seasonal bouts of intense transmission. And so we wanted to check whether the complexity of infection and the level of inbreeding of the various infections are similar across the continent. When we looked at the genomic data from 2,300 parasites collected from 15 countries and 23 endemic communities, we could clearly show that there are clear differences in the level of inbreeding of the parasite in the different communities. There were some countries like Ethiopia and Madagascar which showed significant amount of inbreeding of the parasites in those communities, while most of the rest of the continent did not show that much inbreeding. Indeed, you could actually see up to 9 infections in some cases in Ghana and in Malawi, infecting a single individual, showing that this parasite has adapted quite well to persist in the human population. Now, the second thing we wanted to check was whether the parasite within Africa are similar. And the reason why we're doing this is because in previous studies that have been published from the genomes that we had generated, together with genomes contributed by other partners in malaria endemic regions, and these analysis was done by the malaria gene. What it showed is that the African population was mostly one single ancestral entity, with the exception of some of the parasites that were collected from Eastern Africa. And this is clearly shown in this PCA map here. So what we did in this study is to exclude most of the parasites that come from the other parts of the world where malaria is endemic, and focus only on the parasites from Africa and do the analysis. We actually tweaked the analysis to include slightly more sensitive approaches by measuring, for example, the parts of the genome that are preserved between different parasite by pairs of parasite isolates, and the parts that are diverse between the parasite isolates. So combining similarity as well as diversity matrices using chromosome painting, what we succeeded to do with this analysis was to separate the parasite populations we are analyzing into 6 major groups. There is the main West African group composing of most of the isolates that we collected from West Africa. There is the one that comes from Central Africa. And then there's a group in Congo, which I thought would belong to Central African countries like Cameroon and Gabon, but it was separate from the rest. And then there is Ethiopia, which is a highly divergent population, completely different from the axis of divergence of the population from east to west. And then there is as well the populations from the southeast, which constitute those from the east, that includes Tanzania and Kenya. And then the southeastern population, which included Malawi and Madagascar. Surprisingly as well, the population from Madagascar, which is an island that we thought would have been as divergent as that from Ethiopia, wasn't divergent. And the indication that most of the infections that moved to Madagascar came from mainland Africa. Now, looking at this data, we wanted also to check whether these separate parasite populations are actually unique entities or they are sharing genetic material. And one of the ways that we did this was to quantify the amount of genetic material in one population that we modeled to have come from the other population, from the other subpopulation. And so for our 6 subpopulations, what we could see in that analysis, that when you look at parasites from West Africa, a lot of the infections there, they mostly share their genetic material with those that come from Central Africa. While those in Central Africa will actually have material that would have come from West, as well as from the eastern part of Africa, mostly from Congo. Congo itself had material almost from all populations, and there seems to be more sharing of genetic material as you go eastwards to the eastern part of the continent. Hence, you would have, there seems to be a lot of exchange of genetic material from parasites that are in the eastern part of the continent as compared to the western part of the continent. Now, Congo in particular was an intriguing population to us because it seemed to be a central population most of the time, separated from the east, west, and central population, as well as from Ethiopia. And when we try to quantify, for example, the amount of genetic material that Congo has that is sharing with all other populations, it seems to be the only population that had a minimum of about 4% of genetic material that is similar to those that are coming from several other populations across the continent. And these of course, would draw into some hypotheses about the origin of malaria in Africa. The origin of malaria altogether, especially Plasmodium falciparum, which is the malaria parasite study. Now, there have been several hypotheses about this origin. And one of those has been that they came from the great apes. But the question is, which of the great apes did it come from? Is it the gorillas, is it the chimpanzees, or the bonobos? Now, the range of distribution of the gorillas, the chimpanzees, and the bonobos is shown here on this map to the left. The gorillas are more found in the central part of Africa, while the chimpanzees and the bonobos are also found mostly in Congo. But the chimpanzees are found in the western part of Cameroon, where some studies have said that's where malaria actually jumped into humans. But there are also western chimpanzees in the southern part of Cameroon and Gabon and Congo. There are also gorillas as well in Uganda, in between Tanzania, Uganda, and Kenya. So there are several great ape populations in Africa that could have actually been responsible for the jump of the malaria parasite P. falciparum into the human population. So what I've done here, looking at all the literature, is to look at our data and also look at the distribution of the vector species. You see that one of the most efficient vectors that exists for malaria, it has a distribution that is quite similar to the distribution of the great apes. And so it looks like the hypothesis that there has been a jump driven by probably these vectors from the great apes to humans might be possible. But once again, which of the apes and where did it happen remains a question that several studies have been trying to pinpoint. And there is a narrowing of evidence that the jumping might have happened from gorillas to humans, and that this event might have happened only once. Whether it happened in Cameroon, where I originally come from, or in Congo, also remains a question. Now, we are doing some further analysis of the data that we have, and there are some indications that the population that is in Congo seems to be the population that is carrying a lot of the ancestral haplotypes that are present in all other populations. And if that be the case, it would be similar to what we are seeing in the human population, where the population that is most ancestral, which is the ones in Africa, they're carrying a lot of the haplotypes that you can see in all other populations. So there is a lot of diversity that is present in Congo. And you can see from these new analysis that is being done by Martha, who is present in our group here in MRC, this unpublished work, we can see here that the Congolese population has parasites that can fit in several of the different groups that we have previously identified with SNP data. So again, this supports some of the proponents. There was a perspective that was written by Carol when we published the paper on SNPs that was suggesting that the most likely event of zoonosis that led to the spread of malaria in the human population occurred in the Congo, and then these parasites spread to the rest of the continent. From these new sensitive analysis we're doing with microsatellites as well, what we've seen is that we are able to split the population from Malawi into two populations. There is one population that's quite similar to the population in Eastern Africa, that includes Tanzania and Kenya, but there is a population that is unique of its own. That population is similar to the population from Congo, and they form a cluster of their own in this PCOA plot that we are displaying here. So again, these put together continue to support the fact that from doing this whole genome analysis, being SNPs or microsatellites, we are driving to an evidence of an event that occurred with the parasite jumping from the ape to the human population. And one of the mutations that would have allowed this to happen would have been mutations that occurred on the ligands of the parasite, on the surface of the parasite, on the surface of the parasite, where we have ligands that bind to different receptors in the red blood cell. So it is being hypothetically suggested that the mutations occurred on some of these ligands, and that these mutations allowed the parasite to be able to bind to receptors on the surface of the human red blood cell and then invade the red blood cell. So parasites from the great apes, once they mutated, they get transmitted by the mosquitoes, and then they invaded the human population, and then adapted in the human population and started circulating within the human population. So together with one of the fellows in our group, one of the things we want to check was that this hypothesis of mutation of the parasite ligands and then the jumping into an attachment to receptors actually holds true from the data we've collected across the continent. Now, this work is being done by Fadel Diop and has not been published yet. And I'm just showing an excerpt of what we've seen from the analysis. Now, when we look at diversity of genes across the parasite, and this kind of diversity we're referring to is diversity where we're looking at mutations that would have occurred between the type of the gene in the P. falciparum population compared to the type of the gene that might be present in the parasite that are collected from grape apes. Now, when we do this analysis, there are several regions that we see very strong emergence of diversity within the parasite population, the P. falciparum population, in all the different subgroups that we had previously identified. Now, one of the strongest regions there is one that codes for a surface protein called a Duffy-binding protein ligand 2, and that is coded for on chromosome 10. Now, in these analyses, it is highly diverse. You can see it in all populations, except in one population, the population from Congo. So once again, this is only speculative and suggestive that one of the genes that actually allow the parasite to be able to jump into humans and become a parasite of humans is a modification and mutation that occurred in this gene called DBL-MSP2. Now, DBL-MSP2 is one of the genes on the surface of the merozoite that interact with proteins on the surface of the parasite and together form the complex that allow the parasite to invade the red blood cell. Now, in the work that we're doing, looking at the data collected from the various parasites across the continent, we have structurally modeled the DBL-MSP2 gene, and we are showing that the part of the gene which has the strongest diversity, has the hottest spot of diversity, this reddish bits of the model is the region of the gene that actually binds to the receptors on the surface of the red blood cell. Again, kind of culminating the evidence that mutations that occurred here and the diversity that occurred in this region allows the parasite to adapt to different human populations and be able to spread across the entire continent. The flip side of this is there are also sections of the genome that are conserved, which means they're having similar haplotypes shared across several populations. And what we've done here is to try to look across the populations, to look at identity by descent, and where we see high proportions of identity by descent between parasites from different populations across the continent. Different from what we've seen with the genes that I showed, like the DBL-MSP2, that are mostly surface proteins. When we look at these different type of analysis and look at conservation, what we see here are genes that seem to be related to more recent events, like genes that have allowed the parasite to survive against drugs. And an example is the chloroquine-resistant transporter coded for on chromosome 7, or the dihydrotaurate synthetase coded for on chromosome 8, that is a marker of sulfadoxine resistance. We also found some other genes like the amino acid transporter on chromosome 6 that shows very strong conservation of haplotypes. That's an evidence of a selective shift that is imposed by a recent event like the use of drugs. But this gene, as of the time that we did this publication, had not been shown to be responsible for drug resistance or being associated with drug resistance, even though it has been seen in the company of the chloroquine-resistant transporter in most of the genetic studies that we had done. So we believe that the amino acid transporter on chromosome 6 from this evidence might be one of the genes that are contributing to drug resistance indirectly, probably in epistasis with chloroquine-resistant transporter. And there is more recent evidence that is showing that it's actually doing so. Now, besides these known drug-resistant genes, there were also some new regions that we've identified from doing this continental analysis, like the case of a region on chromosome 12 that codes for a protein that has recently been found to have mutations only in patients that do not clear their parasites after treatment with admixed combination drugs. And these patients were all African patients. So it's possible that this region that we're seeing is the emergence of a new region in the genome that is contributing to the development of resistance or tolerance to the new drugs that are being used against malaria. Now, this is not our study, but further to these analysis and other work that has been done, there's been some forward genetics work that has showed that some of the mutations that are present in this region, actually, once you modify the gene and include this mutation in parasites, wild-type parasites, it increases their survival against the current drugs that are being used. And this work has been done in vitro in the group of Colin Sutherland in London. So very clearly, by using genetic data and by combining data from across the continent, what we seem to see is that there are signatures of adaptation from recent events like the use of new drugs that could be identified and that are important for us to continue to monitor, as these may help us to better preempt the emergence of widespread resistance like had been seen against previous drugs. So one other thing that we thought would be important as seeing all those conserved regions and the emergence of new regions is to look into the parasite genome and exclude all other regions of the genomes that have no direct or suspected connection with drugs, and interrogate the structure of the parasite population using just the. And the question here would be, do different parasite subpopulations from West, Central, East Africa, or from the Horn of Africa have a different backbone of drug resistance? Because the patterns look quite consistent, and it looks clear that they're using similar mechanisms to actually fight drugs. But when we put these data together and do the analysis, what we see is that the parasite populations that we have are actually 3 subpopulations rather than the 6 subpopulations that we had previously described. Now, these 3 subpopulations have a population that includes almost members from every different part of the continent. But there are some that are actually kind of restricted to different parts of the continent. Like in West Africa, for example, there is a subpopulation here that is dominated by Cote d'Ivoire, Gambian, and Ghanaian isolates. So they would seem that there is a backbone of drug resistance that is very Western to Africa, that's common to West Africa, with a bit of infusion of parasites that might have migrated from Central Africa to West Africa. Now, this kind of reminiscent of the history of drug resistance, where drug resistance to previous drugs are most often come into the eastern part of Africa, where you have the parasites mostly belonging to this group here, and then they have flown to all other parts of the continent. And that's why the eastern parasite population belong to this core group in the center. Now, we have repeated a similar analysis that we did with the rest of the data from the genome by asking what is the proportion of drug-resistant haplotypes that are shared between the different components that we've seen here, the different subgroups that we've seen here. And essentially, it looks very similar to what we had expected and what I just described, that the eastern populations seem to share and the central populations seem to share a lot of the drug-resistant haplotypes with the West, even though there was a lot of Western haplotypes that were actually mostly located just in the West and not shared with the rest of the continent. And that's quite important because it indicates that there is the possibility that parasites in the continent have evolved drug resistance on separate backbones. And this would change the way that they actually respond to drugs differently. And we are seeing this happening now to the new drugs that are being used, where there are cases of emergence of drug resistance in the eastern part of the continent, in Rwanda, for example, and an indication is occurring in Uganda, and probably also in Tanzania. Now, we've not looked across the rest of the continent intensely to see whether this pattern is this pattern of local emergence of drug resistance is common. But we know quite well that for one of the drugs, like the sulfadoxine and pyrimethamine that were used in combination, the haplotypes that are contributing to drug resistance, there were some indigenous haplotypes already circulating in West Africa before the main haplotypes came from the East. So this is an analysis, again, that I'm showing you from the work that's being done by Martha, just to map the major traffic lines of parasites across the continent. And what we see here is that there's a lot of traffic of parasites in the eastern part of the continent, which will support the fact that when a parasite that emerges with resistance occurs in Congo or from Rwanda, for example, this could rapidly spread in the eastern part of the continent compared to the western part of the continent. But there is clear connectivity between almost all malaria endemic regions, fueled by probably human travel that is increasing across the globe, across the continent most of the time. Now, this does not only affect drugs, because there is also the sharing of alleles that are being used in the current vaccines. So there's the possibility that this movement of parasites between different parts of the continent would also allow parasites that carry vaccine-resistant alleles, for example, to circulate and then expand in other countries across the continent. And so it's actually important, beyond just doing these large-scale studies that I've showed here, that we should focus on doing studies in single countries and monitoring the parasites as the various interventions are being implemented. Now, most of the NMCPs across countries, they're following the strategy that the WHO has recommended, and they're going for elimination of the parasite across the continent by ramming up interventions using mass approaches, such as the use of SMC, which is a chemoprevention, based on SP and amodiaquine. I mean, SP and amodiaquine, there is already resistance to SP and amodiaquine, and we know that already. So the mass implementation of this is further selecting for parasites, and the movement of people will be circulating these parasites across the continent. So it's important for each country to actually focus on monitoring the parasite population in more detail to understand how they're evolving and whether the patterns that are being recognized continentally are persisting within populations, or they are degrading within their populations, or spreading within smaller populations in country or in region. And as we say, charity begins at home. Some of the work that we're doing with these small country populations has started where I am actually resident, at MRC in the Gambia. The Gambia is the smallest country in the mainland African continent. It is at the western tip of Africa. We call it the gateway of Africa, or the smiling coast of Africa, because of the Gambian River, which is the beautiful river. The MRC is located not very far from the Atlantic Ocean, actually. And so it's a fantastic place to work in. And there are several different projects going on, both research and intervention projects going on in the Gambia on epidemiology, clinical trials, vector biology, and of course, genomic surveillance, which is what I am mostly doing. So luckily enough, the Gambia had the MRC, which had been here for several decades. I mean, the MRC will be celebrating the 75th anniversary this November. So it's been here for several decades. And over time, there's been several collections and surveys and clinical studies that have preserved blood in the biobank at the MRC. And we've gone into the biobank to look for the specimens that we could pick as far back as 1966. So we have now sequenced parasites from the Gambia as far back as 1966 and parasites from several different locations for many different years across the country. Now, what we are saying with that data is that we can start to use that data to actually show that there is a decline in the prevalence of infections in the Gambia. Already, this has been reported a lot by the NMCP, and this map that I'm showing here show that the transmission of malaria across the Gambia is quite heterogeneous. Malaria remains in smaller pockets. There are hotspots of transmission in some villages and a lot of cold spots across the continent. I have lived in the Gambia for 16 years, and in my 16 years of life in the Gambia, I've never had an infection of malaria from within the Gambia. So it's clear evidence that and I live in the western part of the Gambia. It's in Banjul here. The MRC is located here. So there is very low density of infection in this area, and that's probably why we haven't had infection. But we have field stations in the eastern part of the country where we go and work, and there is a higher prevalence of infection in that population. And we can see the difference by just looking over time, by just looking at the genetic data that we have collected from the various parasite population. So as we did with the African population, and very early on in our studies, what we wanted to ask, especially because I had an interest in drug resistance, was to ask whether the markers of resistance that are continental, as described in Southeast Asia and other populations, are also present in the Gambia. And what we've seen by combining some of the samples that we sequenced with those from Senegal, because the Gambia is actually within Senegal, is that those regions of the genome that show selective shifts due to the use of drugs remain quite consistent with what is present in most other populations, including Southeast Asia. But there are others that are also present there that have not been properly described in other populations. Now, the strongest of these signatures, of course, is the one that I said is present on chromosome 6, and it's around a gene that codes for an amino acid transporter that we suspect might be a gene that is cooperating with the chloroquine-resistant transporter to exacerbate chloroquine resistance within our population. Now, we do have evidence of that now in the paper that we'll publish very soon, but I'm not going to present that. I'm not boring you with that presentation, which is quite detailed and includes a lot of experimental genetics work as well, beyond just the population genetics work that I'm presenting here. Now, following on from that analysis, I had focused a bit on the amino acid transporter just to use slightly less technical and less intensive methods to prove that this signature of selection is a signature of selection that has existed over years, similar to the signatures of selection that have been imposed by drugs on chloroquine-resistant transporters and other genes that are associated to drugs. And what I'm showing here, for example, is what happens when I did the analysis with microsatellites around the region of the amino acid transporter between the year 1984 and 2014. This spans about 30 years. And you could clearly see from these analyses that over time, there is a buildup of similar haplotypes around the gene. And this definitely supports the hypothesis that Sarah Vogtman did propose here, where there is an emergence of an advantageous mutation. Over time, the haplotype on which that mutation emerged will become the dominant haplotype in the population. So by doing this work using microsatellites, we've clearly shown that amino acid transporter over time had been selected similar to drug-resistant genes in our population. Now, this is a combination of all the populations that we have, just showing where you have the strongest signatures of selection and adaptation. And what we clearly see here is that the drug-resistant genes are the strongest. But there are others that we haven't described beyond the amino acid transporter. For example, this region on chromosome 7 and many other regions across different chromosomes that I'm showing here. Now, to exemplify these to the drug-resistant genes, what you will see here is that we take most of the haplotypes that we know have the strongest association with drug resistance. You see over the 30 years, the increase in the frequency of the haplotype to almost fixation in the population. Now, this is obviously quite important to us, and it's something that we are following up and trying to link this to the resistance of the parasite in our population, and to further understand the role of new genes that we are identifying by this genetic work in enabling the parasite to adapt to drugs. So an example again of what I've shown again is identifying one of the alleles that is a mutation on one of the alpha helices of PF81, the amino acid transporter, trying to follow it over years. And what we see here is the growth of that allele over time. And this, again, as I said, hypothetically parallels the growth of the resistant allele to chloroquine, the allele that is strongly associated with resistance to chloroquine. The structure of these proteins and the structure of the chloroquine-resistant transporter are quite similar. And put together, it would seem that definitely in these analyses and further analysis that we have done, that this amino acid transporter is one of the genes that's contributing to the survival of the parasite against drugs such as the chloroquine-resistant transporter. So to take this further and just to push to wrap up, and I'm going to be very quick about this, is trying to understand how these alleles that we've identified that evolve over time, how they might be spreading between populations. And to do that, we kind of use genetic data to do kind of a transmission analysis, similar to generating transmission networks, and to understand where the hotspots of infections are and where the hotspots of these various genotypes that we've identified might be in our population. Now, this is an example of a small study in the Gambia that could be expanded to other populations across different countries in Africa. And so what we did in the Gambia, obviously, is to look for several villages and just collect parasites over time and over space. And then went for to do the genetic analysis using SNPs, either the whole genome SNPs or using a subset of SNPs in the genome to understand the relationship between the parasites at the time that we collected, the place that we collected, and the place that we collected. So just looking at that data, one of the things we did to make this possible was that we developed a model that allowed us to actually infer the transmission cycle that may occur from one individual to another individual, or a situation where two individuals are affected by the same individual. And in that, following that analysis, what we could show from that analysis is that the unit of transmission of infection actually is the household. So most of the infections that you see in our population are occurring because someone in the household is transmitting the infection to the next person that share the same house. And you could see that from the household down to villages that are distant apart, the similarity between parasite infections completely declines. And this is the same thing, but less so when the parasites are separated by several days. Now, clearly, the infection unit is the household. And it is important for the malaria control programs to know this because it can help them to use new approaches, such as reactive household intervention, where if you identify an index case, you can then move forward to treat all the members of the household. Now, an example here is this is how houses in some parts of rural areas, not only in the Gambia, but parts of West Africa will look like. So if one individual is infected here, the vector will likely move the infections across the entire members of the other houses in the compound. So infection starts within the household and then they spread within the compound. So when you look at compounds that are different from each other, separated from each other, you will see a significant difference in the amount of genetic material that is shared by pairs of parasites that have been actually isolated from infected individuals in these compounds. And so this kind of information is information that we shared with the National Malaria Control Program to help them to better refine their strategies. The second information we get from doing this genetic analysis down to the level of the village and not just the country is that we are able to identify within different villages individuals, this was prior to COVID, that we call the super spreaders. And here we're showing from several villages that we have super spreaders. Village K, for example, had a high number of super spreaders that these are individuals that share infections to more than 2 persons. And in some situations for Village K, for example, there is an individual that probably likely shared the infection to 10 others in that village. And this name super spreader, we find it before COVID came. And again, a similar analysis had been done in COVID and it's showing similar patterns. We're furthering this analysis. Now, this also has not been published, but we're furthering this analysis by trying to find out to quantify the amount of parasites that flow between villages, as well as the amount of parasites that flow within villages. And here we're just showing the connectivity between those parasites that we identify between villages, as well as within villages. And what the National Malaria Control Program needs probably to do is how to break these transmission that occurs within villages and between villages but probably apply new mass treatment approaches or reactive household interventions to stop the transmission flow in the country. And lastly, as we would like to always look at adaptation, the question we would like to also to ask is, do these movement of parasites within compounds between villages move along with parasites that carry drug-resistant alleles? So we're showing here again that using the genetic data, that there is movement of parasites across the country, a movement between different villages and different parts of the country, with drug circulation most commonly spreading within villages rather than between villages. Again, this is information that needs further verification with more data, and hopefully we're going to get more data to continue to look at this flow of parasites and the flow of the drug-resistant genes and the drug resistance markers, both old and new ones, that we are identifying from our analysis. Now, put together, what this analysis of continental and country parasites is suggesting is that we do have communities of parasites that exist across the continent. And even within specific communities in different regions where we have subpopulations, there are common markers, but there are also specific markers in some populations that need further investigation. Now, those specific markers, like the markers that are emerging in Southeast Asia, are emerging on a backbone that can be shared across the continent. And this will facilitate the development of tolerance to drugs, vaccines, or just increase the likelihood of the parasite being transmitted by different mosquitoes across the continent. So this altogether shows that studying genetics and looking at the genetics of the parasite is important for us to do and to continue to do to support the strategies that WHO has put out and the work of the National Malaria Control Program. Now, we can make some recommendations for this, that the National Malaria Control Program obviously should be part of this kind of work. It shouldn't just be work by the scientists. It should be work within the partnership between the scientists and the National Malaria Control Program, with the National Malaria Control Program playing a significant role in helping us to refine our surveillance approaches to better match with their interventions, and that will help them to better implement interventions or have more effectiveness in the interventions that are being implemented. Now, a lot of the work that I've presented here is not done by me alone. It's done by a lot of people. I mean, this picture is slightly older than the picture of the group that I have now, but it has most of those that are working with us. Analysis that I've shown have been done by Fadel, Martha, Karim, myself, and many others here have actually contributed to a lot of the things that we do here as a group. And we do it because we have fantastic partners like the Malaria team that has provided genetic data to us from the Sanga Center. We have the PDNA where there's harmony in partnership across the continent, and they've helped us to get data from across different groups from different countries. And that has allowed us to be able to do these analyses that are presented here. So I'm thanking all these different collaborators and funders like the MRC as well, and my boss, of course, at MRC The Gambia, and the statisticians, and in the participants that have provided their samples to allow us to do most of the work that I presented to you here. Thank you very much. I hope I've been on time. That is what I had planned to show to you today. Thank you very much, Tina. Over to you.
Thank you so much, Dr. Alfred, for such an interesting presentation. This is really amazing work that you're doing towards the efforts of eliminating malaria on the continent. You're right on time. I think we've got about 12 minutes for questions. So I'm going to open the floor for questions. If you have a question, you can raise your hand or type in the chat box and I'll read it out to Dr. Alfred. First, we've got Wisdom. Wisdom, you can go ahead and ask.
Thanks very much, Nakao. Great work, Prof. Amambuangwa. I just was wondering that the work on vaccine for malaria has been long, and I presume that much of the failure of that has been because of the diversity of malaria species and this genomic diversity too. How likely is it that this vaccine will work in the midst of all this diversity across the continent?
Yes, thanks very much, Wisdom. That's an important question. Now, so I have not presented any diversity on vaccines at the moment, but you're quite aware that there's some work we've done, including work in Ghana, that has shown, for example, that the circumcellular protein that is used in the RTSs is quite diverse even within Ghana itself, and these changes over time. That said, there is something that we call strain transcending immunity. So the vaccines that are currently being generated can allow for a good number of parasites of different allelic types to respond to the vaccine. So they are effective to an extent, but the diversity has been a problem. And that's why the RTSs up to the moment has only an efficacy that is not wider than it's not up to 50% in most populations. And so it's possible that you will get some protection from the vaccines that are being used. And using this kind of data, new targets of vaccines are being identified. What I've not shown here is some of the analysis that we've done to show which immunogenic genes are highly conserved across populations. But there do exist some genes that can add to the pool of genes that are currently being developed for vaccines. So yes, diversity is going to be a problem, as has been the case for COVID, as has been the case for several different organisms. For pneumonia, for example, they're on PCV 13 or whatever number it is. 13 is a valency of the vaccine. So they include 13 different serotypes in a single vaccine. So that's something that is possible to do in malaria in the future, at least with combining the vaccine and other mass treatments. It's probably a mass preventive tool. It might be possible to bring down the rate of malaria beyond what is currently experienced. So I don't know what I can fully answer your question. I hope I've been able to answer in some way. Thank you.
Yes, thanks very much.
Thank you for that. Are there any more questions?
Yeah.
You can go ahead.
Okay. Thanks very much, Prof. Alfred, for the wonderful presentation. This is Francis. So actually now in Africa, we are actually seeing a slow decline in the 76T mutant allele, which is associated with chloroquine resistance, and noting that chloroquine resistance is no more in use. Do you think that the AAT1 mutation may be contributing to this slow decline in the 76T mutation?
Thanks, Francis. It would seem that PFAT1 and PFCRT are in epistasis.
Okay.
It would seem that you can check bioarchive. We have a manuscript in bioarchive, which is under review by Nature Microbiology, that is showing strong evidence that there's linkage disequilibrium between the amino acid transporter and the chloroquine-resistant transporter, despite the fact that they are on two different chromosomes. So it would seem that there is some dynamic between the two variants. Now, but across Africa, there's something that intriguingly is happening. In the eastern part of Africa, with the withdrawal of chloroquine, what has been experienced is a rapid reversal of the 76T mutant form, almost the complete disappearance of that form from that population. Meanwhile, in the Gambia, for example, I mean, you can see from this plot as recently as 2015, and at the moment in recent data, we're still having over 50% of the parasites having the 76T mutation. And this type can hypothetically be due to the fact that the 76T mutation in West Africa is not as deleterious, because the compensatory mutations present in the West African genome backbone of the parasites allows the mutation to persist even in the absence of the selective pressure from chloroquine. So it's something that is important to, obviously, to study and understand the dynamics, because withdrawal of drugs, the expectation would have been every parasite would have reversed to wild type, and then people could start using chloroquine again. But that's a risk that we cannot take, because if you do that, there's a likelihood that there will be rapid selection back to the chloroquine resistant type in our population. Thank you. I hope I've been able to answer, Francis.
Yeah. So if the moderator will allow me, I have one more. Okay.
Francis, you can go ahead. I'm going to limit the questions up until Blessing. We've got 2 hands raised and also a question in the comment section. So after Francis, Dominic, you can go. Thank you.
Okay. Wow.
Thank you.
Thanks. Yeah. So you actually showed that there is a distinct Ethiopian parasite population structure with respect to your microsatellite data. Could you comment if if Plasmodium vivax and also Anopheles stephensii, I mean, what are their impact on this population structure? Because stephensii is actually, I mean, at the Horn of Africa, and also vivax is like also there. So what could be the impact on the PF structure with respect to other parts in Africa?
I think, yes, thanks, Francis. I think there are 2 kinds of structure here. I think what we've shown using chromosome painting actually relates more to an ancestral structure of the parasite. And these probably are things that would have been events that drove these divergence between the subpopulations would have happened probably before the appearance of stephensii in Africa. So something else would have led to that. Now, we had suspected when we were doing the analysis that the divergence of the Ethiopian population might have been to the fact that it is sympatric with P. vivax in the Horn of Africa, and that would have actually led to some kind of differentiation from the rest of the African population. Yeah. But that seemed not to be the case, because P. vivax is also present in Madagascar, but the Madagascan population was clearly a continental population because they were quite similar to those from Malawi. So P. vivax is not the contributing factor. So what we would suspect further, I mean, Lemo can do this further in his analysis currently of malaria parasite populations from Ethiopia, is whether there's a barrier between the parasite, the geneflow, between African continent and the rest of Ethiopia. There is some limited amount of it, which we did show with our analysis by chromosome painting. And a second hypothesis is that some of the parasites we see in Ethiopia could also have been parasites that actually came through from Southeast Asia and through the Arabia. So that combination between African populations and the populations in Asia and the Arabian Peninsula might have created a subpopulation in Ethiopia that is unique. That's hypothetical, because we don't have the data from Arabia to do this analysis. But obviously, there's data from Asia that can be studied further with the data from Ethiopia to see whether there are some sharing of genetic material between these 2 populations. I think more recent structure, it could be driven more by drugs that we're using and other interventions, but that doesn't seem to be what we've shown here. We've shown something slightly more ancestral than due to current events. Thank you, friend.
Thank you.
Thank you for that. We've got about 2 minutes, but I'm going to extend it by an extra 5 minutes, if that's okay with Dr. Alfred.
That's very fine, Doctor. Please do.
Okay, thank you. There's quite a number of questions coming in, but I'll go to Dominic to ask his question, and then we'll proceed to the other ones.
Yeah, thank you, moderator. Prof, that's a very remarkable work. The data that you've shown here shows how great and important the importance of research work in getting research to be translated into policy, the population. I also noticed that you were able to use data as far back as 1960s and the 1980s to the present, which is very remarkable. My question is, will you be able to use this data to predict future genetic diversity and gene flow, such that we might be able to design drugs using that information or vaccines in the future? Because as we all know, the parasite keeps, and it looks like it's trying to be ahead of us. Is it possible we could use the trends and information that you have over the years to model something that can predict targets for the future? Thank you.
Yeah, thanks, Dominic. Yes, evidence modeling, Dominic, I think modeling is possible, but whether the current data can be predictive of what will happen next in terms of the genetics of the parasites in the populations, I can say there is a bit of shadow there. We don't know yet, but it's possible. As you are well aware, already there are several models that predict the likelihood of emergence and spread of resistant mutations in parasite and vector populations, and similar methods can be applied. But the current data can also be used as well as a background to check whether those models do exist. If a predictive model is done using a complete simulated data, that can be checked with the data that we have temporarily to see whether the model that has been generated maps to the evolution of some of the alleles like PFCRT and PF51 that we've shown here. So that these can contribute to that, but it will require significant thinking around mathematical models that can predict events that we have hardly any control of, because there are so many different factors that are determining not only the ecology of the parasite, but the evolution of the parasite in the population, including interventions and demographic of the host, human host, as well as the vector host and the various subtypes. Yeah. Thank you, Dominic.
Thank you, Prof, and keep up the great work.
Thanks.
All right, thank you. I'm going to read a question from the chat box posted by Kevin Eso. Thanks for the excellent talk, as usual, Dr. Alfred. A recent paper on human evolution by Mark Lipson et al. From this year found that the Duffy-negative alleles that confer resistance against Plasmodium vivax were already fixed in ancient genomes from Cameroon, about 8,000 years old, while they were completely absent elsewhere. I'm just wondering what you think this tells us about the origin of malaria.
All right. Yeah. Thanks very much, Kevin. Now, there are two groups of malaria parasites here. I think what I've presented here is the most common Lavernae group, Lavernae parasite, which is P. falciparum. Now, there is a P. vivax group, which they seem to have been slightly older. It's called an old world Plasmodium species. So it's possible that the event that led to the jump of P. vivax to humans occurred in Cameroon, around Cameroon, and occurred earlier than the events that led to the jump of P. falciparum to human populations. Put together, the hypothesis about the jump of parasites to humans is linked to the human migration and development, where, for example, when farming started, for example, in Africa, and this is beyond 8,000 years ago, and there was the presence of water that encouraged the vectors and different types of vectors to propagate across several populations. And human populations started to expand, and then there was more contact between humans and ape species, and that allowed for the jump of the parasites from apes to humans. So yes, what you presented does seem to suggest that the vivax species might have actually jumped into human populations in Cameroon, but I don't know whether this actually is the same situation for Plasmodium falciparum, because as I said in my presentation, there are two schools of thought that seem to be emerging. One that thinks that there was a jump event that occurs from the Western highland gorillas in West Cameroon, what we call Southern Cameroons at the moment, into humans. And it's a possibility that the Western gorillas in the southern part of Cameroon and Congos might also be responsible for the jump into the human population. So all of these are possibilities. But what we are showing with the data here for P. falciparum, obviously, is that the most ancient and population which has most of the genetic material that is shared by all other populations seems to be the one from the Congos. And these samples were collected from the western part of Congos, where we have a high density of the western lowland gorillas. So there's a likelihood that these might have happened in the Congo as well, but it's up for investigation. Thank you very much.
All right, thank you for that. We've got one more question and 1 minute left on the time, so I'm going to indulge Vincent Duru, who's saying, Thank you, Dr. Alfred, for the wonderful presentation. My question bothers on the effect of climate change in the transmission patterns of these MPs. What do you think the association between this climate change and drug resistance will be within population and between populations in Africa?
All right, that's a slightly complex question there, Vincent, and thanks for that. I mean, climate change, the simplest thing we can say is how climate change is going to affect the malaria vector, parasite vector, which is the anopheles species. You might have an increase in vectors, you might have a dominance of a single vector, and hopefully not the most efficient of the vectors, which is loci. If that happens, obviously that would affect the rate of transmissions across the continent. Now, climate change might also result in populations becoming less adherent to control interventions, such as the use of bed nets because of increase in temperatures across the continent. If there's an increase in temperature and people stop using bed nets, obviously they'll be more exposed to vectors. And if that be the case, you'll have issues. So I see two things here: changes in demographic density of the vectors that may affect human behavior as well, and that would probably lead to a reversal in the course that we're seeing in malaria elimination. And there are many other possibilities that climate change might actually cause that I am probably not an expert to be able to expound on. But these are things that are interesting, obviously, for us to look at together as a scientific community in Africa trying to contribute to malaria eradication. Thank you.
Thank you so much, Dr. Alfred, and to all the participants, thank you so much for engaging. I think this is a very interesting talk that has a lot of interest from people that joined today. Just to note to everyone that the recording will be made available on our YouTube channel, H3A Bionet, and it will also be available on our website. I think it's been a great day for this presentation. I'm just going to read a comment from Sainabo. I hope I said that right. Forgive me if I haven't. Dr. Alfred, you have been doing and continue to do a significant about science for malaria, as far as malaria parasite biology goes, looking at drug resistance. You could on the presentation, but it looks like it's a question, so I'm going to leave it there. I thought it's talking about the impact of your work that you're doing in malaria. Thank you so much for the presentation, and that's it from me. Thank you everyone for joining. If you have any closing remarks, Prof. Alfred, you can go ahead and share, and then I'll close up the meeting.
No, thank you very much for the invitation. I think it's great to do this presentation, and I'm happy that it's been well appreciated. And hopefully, if anyone has any more questions, please feel free to send me an email. I'm happy to have a chat and or to respond to you by writing. Thanks a lot.
Thank you, everyone. Enjoy the rest of your day.
Thanks. Bye.